Welcome to my world. Another day, another time. This is another episode of Knowledge Acquisition. We shall be talking on the outbreak of meningitis outbreak. What does it mean? You hear so many times, and most of these times are things that are laboratory based. It takes the lab to really confirm an outbreak. But normally, you want to believe that if somebody has meningitis, it's a serious problem, and do we consider that every meningitis as an outbreak of meningitis because it just happened? Well, no. To get to know more of these times, up to what is endemic, epidemic, and pandemic, and other terminology like that, I want to expose them using the laboratory processes to get at these various terminology. So let us now move on to the slide section and experience more knowledge. If you like what you listen to, press the like button, subscribe, and you'll be notified every time the video is out. Thanks. Meningitis outbreak. What does it mean? Yes, that is what we are out to explore. Let us start with outline. Definition of terms, type of meningitis, the laboratory processes, and finally, conclusion. Meningitis is a serious infection of the meninges. The meninges is the brain covering the brain and spinal cord. Children from 4 months to 1 year are mostly affected. Several different bacteria can cause meningitis, but the commonest ones are Streptococcus pneumoniae, Haemophilus influenzae, Neisseria meningitidis. They are transmitted from person to person through droplet of respiratory or throat secretion from infected persons. We are now more or less talking about the outbreak. The outbreak is a meningitis. Meningitis outbreak occurs when multiple cases of the same serotype, whether biotype or even the character, happen in a population over a short time period. Outbreak can occur in communities, schools, colleges, prison, and other. The fact is just that one strain is being replicated over and over. Endemic. An endemic disease is a disease that is always present in a population. Epidemic is defined as an outbreak of disease. Also, it's an outbreak that spread quickly and affect many individuals at the same time, going out of proportion to adjoining places, even spreading to almost the whole region. The epidemiologists will be looking for the index patient, that is the first patient to be infected, and the strain confirmed, and trace the contact, and they now confirm if the contact is bearing the same, is infected with the same strain of the organism. Pandemic is defined more or less, it is the worldwide spread or outbreak of a new disease. So the pandemic is a global event. It's like COVID-19 moving from one continent to the other. So you are now talking of something that has now spread the same strain, although it could vary by mutation. The type of meningitis are, there is the meningitis itself and a septic meningitis. The meningitis usually is described as bacterial meningitis, which is caused by bacteria and are very pathogenic. That is, the response from the immune system is more of white blood cells proliferations. Viral, fungal, parasitic, they are all under the aseptic. Amoebic, non-infectious meningitis, and drug-induced aseptic meningitis are classified as aseptic. You will agree with me that this kind of list would necessitate different treatments, and so we need to know more to be able to treat right. The differentiation of even organisms that are of the same species. Ordinarily, when an organism species is mentioned, it seems it has same character, but genes mutations necessitate further characterization into strain or biotype, stereotype for study and treatment. Most large epidemics are caused by serotype A when we are now talking of Neisseria meningitidis, just an example, meningococci. But the serogroup C or serotype C meningococci can cause outbreak and occasionally epidemic. That's referencing Wangel and his co-workers. So you can understand that. It is this same characterization that has helped to identify the various strains of COVID-19 viral disease. It is very necessary to differentiate all the different causes of meningitis because the drugs used for treatment are not the same. What kills bacteria are different from what will kill parasites. All organisms are differentiated by their nomenclatures of genus and species, as for example, Neisseria meningitidis, which also has different group and finally strains, have different antibiogram. This also applies to viruses like the flu virus. There is also the hemagglutinin and neuraminidase that are used to differentiate. These antigens often mutate. So the same thing applies, especially those pandemic. Once you have strains that mutate very well, they can spread to all over the world. Laboratory processes you identify involves the surveillance system, which will go about trying to make tracing the thing. Then the samples collected the CSF by lumbar puncture. CSF samples were cultured, and the white cells count are measured to know the proliferation of white blood cells, especially for bacterial infection, and blood culture too. These are in cases of bacterial infection. When we talk of meningitis proper, it could be meningococcal, it could be streptococcal meningitis. Meningococcal are identified by gram staining and by oxidase tests. They are oxidase positive and they are serotyped, determined initially by latest group. Isolates are tested for beta-lactamase. Antimicrobial susceptibility are done because even strains may have sensitivity pattern that is different from the general population, the general group, the genus itself. So for further studies, the isolates identified are transferred to reference laboratory by either lyophilizing it or freeze drying in glycerol broth or glass beads and transported. Typing serogrouping using a ELISA test will help to really further group the bacteria, and the test is very, very useful. All organisms, including viruses, are characterized for full identification. That we must understand because characterization has to do with the gene, the genetic composition of the organism we are testing. So is that genetic composition that tells us more about the behavior of that organism. And you can imagine if it's causing a pandemic or infection, it will be easy for us to produce vaccines from it. Once you are able to get the strain, then you can be able to get specific reactions. The memory cells of the human body will produce and be ready to proliferate immediately. In conclusion, we must talk. The highest burden of disease is seen in a region of sub-Saharan Africa. So it has been demarcated and known as the African meningitis belt, which is especially recognized to be at high risk of meningococcal, especially, but also pneumococcal meningitis epidemics. So often you hear of outbreaks in these sub-Saharan. It is based on the health standard, the hygiene, and the sanitation in all this area, and the closeness, because the infection, the proximity is a licensed vaccines against meningococcal, pneumococcal, and Haemophilus influenzas, and even viruses have been available for many years. We know of the recent one COVID-19 vaccine that people took. Surveillance system activities have been reinforced. That is very necessary if you want to continue to keep in check the organisms that usually produce this meningitis outbreak. Reactive vaccination campaign is prone to help. It will help to minimize the problem. Thanks.